did want to go over some of those commonly missed questions and maybe how we should be thinking through those questions. And that's where I want you guys to be speaking up today. Today's new information is the operon. And so depending on how long we take to get through the recap and the operon, then we have a pretty short follow-up that has a video. The video is 10 minutes, so you probably want to watch that one on your own. And then 2 slides of step-by-step reading, not much, and then 2 slides to label. And there's like 5 things to label on each side. So it's not a lot, but it might happen after class. I'm not sure. So we'll just go ahead and get started and we'll see how the time goes. So here's our test. Woo. First of all, am I sharing a screen that says, here's our chapter 12 test recap? Yeah. Okay. So, so when I look over this, there were some concepts that we knew really well, and then there were some that we struggled with. So there were 3 that less than half of us got right. So your score is dropped down. I dropped it to 26 because that was the high score, but maybe I should drop it to 25 because not even half the people got this 3rd one right. So we're going to look at these 3 questions. So first of all, they all seem to be picture-based. And of course, there was a lot of picture-based on the test anyways, right? So when you get a picture on the test, I want you to look at the picture and analyze the picture before you ever look at the question. What do I know about this picture? What is it demonstrating? What information can be drawn from it? And then look at the question. And again, before you ever look at the answer, choices. Try to come up with your own answer. So what do we know about this picture? This is where you get to talk. If you'd like, you can chat it, but you can also pretend you're in a classroom and voice up.
It has uracil.
Okay, I see this strand has uracil. So that automatically, what is that going to do for me?
It's an mRNA strand.
Okay, so that tells me that I'm looking at mRNA and what process?
Transcription.
Yeah, transcription. Okay, so from this, we got that we are transcribing the DNA and we're making transcription. Okay, what else do you know about? So now we can kind of think about the process of transcription. What do we know about transcription? You're transcribing the genetic code, right? Do you transcribe the coding strand or the template strand? How many people think, you can use your raise your hand if you'd like. How many people think we are transcribing the coding strand? Do you guys know how to do the hand raising and your reactions? Okay, I see 1. Okay. Just so I know how this works, Penelope, since you're camera off, will you raise your hand? I'm just wondering if I do see reactions for people that are faceless. Where'd you go? You keep moving. Okay. Yes, I do. Okay. Thank you so much. Okay. So that was a test. How many people think this is that we code that we transcribe the coding template strand? I said coding the first time. So if you were going to make copies of something, do you make copies of a template or of a code? Which one seems like, if I go to the copy machine and I'm going to make copies of this, does this serve as a code or as a template?
Template.
Yes. So you transcribe the template strand. Remember, because they're complementary, right? So if this is complementary to the template, it means it's also like it's identical to the code. Doesn't that make sense? We're trying to make a copy of the code. We want the code. This is going to be opposite of whatever I'm taking it from. So if I took this from the code, I'm going to get the opposite information as the code. But if I take it from the template, I'm getting the same information as the code because they're both complementary to it. Okay, so that's the first thing. And so if we notice that this was mRNA and this is transcription, then we have to think about which side am I transcribing. And I see that it's complementary to this side, not this side. that means this side is the template strand. This side is the coding strand, and this is the same. So with the replacement of U, that's the same as the code, right? So then we look at our answers, and which one do we choose? Which ones can we throw out? Which ones are simply not related? So also when trying to come up with answers, so we looked at our picture. Let's say you don't know anything still. Now we're going to throw some questions out. Answers out. So which things seem irrelevant or not true? A is definitely not true.
B.
We do read, okay, the coding strand in this process. Because we know that it's the template strand, we can throw out the coding strand. The 3 to 5 reading is correct. You build 5 to 3, but you read 3 to 5. And then 1 and 4. So that would be A and D. So yeah, the anti-sense might have thrown you off, but notice the word template there. So it's so different resources may use different terminology. So anti-sense being the opposite of the sense strand being the coding strand. So the template is what we needed to see in that answer in order to choose C. Okay, here's our next one. What can you tell me about this? If you're analyzing, you just tell me anything you want about this table. When you look at the table and you're all by yourself on a test, you're like, what does this have to do with? So what kind of information comes to your mind when you see it?
It's talking about transcription and translation of prokaryotes and eukaryotes.
Good. Yep. So now I'm guessing I must be doing some sort of comparison, right? So as I compare, how many things are different between the two?
No, 3.
There's 3 things that are different. What are they?
Location, large ribosomal subunit, and small ribosomal subunit.
Good. So so we can see that transcription is happening in the cytoplasm, translation is happening in the cytoplasm, whereas in eukaryotes, transcription takes place in the nucleus, translation takes place in the cytoplasm. So in the prokaryotes, they're in the same place, and in eukaryotes, they're not in the same place. Ribosomal subunits, that's just different sizing. So if we find that our question has something to do with sizing or evolution, that might come into play. Okay, so we're looking for which one talks about the difference in prokaryotes and eukaryotes with regards to transcription and translation. So I'm probably going to not think about the ribosomal subunit, right? The 2 things that were different were the sizes of the ribosome and the location of processes. So which ones of these, because these are all true statements, but which one is relative to that table?
Letter A.
Yes, since the transcription and translation were both happening in the cytoplasm, we can assume they're happening simultaneously, which is something we did learn. In the eukaryotes, they're happening in 2 different places, so they simply can't happen at the same time. Okay, prokaryotes having a shorter RNA strand than eukaryotes, that may be true, but nothing on the table told me about that, right? So if they're giving you a picture, a data table, or a graph, they want the information that you're giving them to come from their resource, okay? So even though these other 3 statements might be true, they have nothing to do with that table, okay? So this is asking about molecules involved with DNA replication. So what can you tell me about the replication process? Maybe you use even like pick words in this picture. How are they used in replication? How is helicase used in replication? Let's start with that one.
Helicase unwinds the DNA sequence.
It sure does. And topoisomerase. Do you guys know what that one does? We didn't really talk about it. Yeah, it keeps it from coming together so they don't stick together. So it holds it open. I can see that I have 5' and 3', so I want to remember that I'm reading 3 to 5. I'm building 5 to 3. That makes them opposite. So if I know that, I know that I'm reading this one 5 to 3, and I'm building my strand 3 to 5. What is this? RNA primer? This is an RNA primer, right? Does that belong there? Yes, it sure does. So the RNA primer there, remember we put the RNA primer there so that DNA polymerase has something to bind to. Otherwise, it's a free end. There's nothing for the DNA polymerase to bind to. So we put the RNA primer down, and then we can build from there. Later, we'll remove the primers. Okay, so we got to think about what all of these things have to do with the process. So let's say I have no idea what's going on. I'm going to narrow down my choices. Which of these statements are simply false statements? So if you're clueless at any point, look at your statements and treat each of them as a true-false statement. So are some of these false statements?
A and B are, right?
Correct. So now the question is, which of the following correctly explains where DNA replication begins? On the strand orientated 5' to 3', reading from left to right. So they're saying reading left to right, 5' to 3'. So it's going this way. So this is the strand they're looking at. So where is the next nucleotide going to go? Is it going to go anywhere in the middle? Is it going to go on this end where it's closed? Or is it going to go right here next to the RNA primer?
Next to the primer.
Correct. So it's going to go immediately to the left of the RNA. Since DNA polymerase requires that. Okay, last one. What am I looking at here? So when you look at a picture, like when you're taking your test, you look at the picture, right away you should figure out, what am I looking at? So I'm looking at transcription and translation occurring at the same time as what I'm seeing in the box. So what kind of organism would this be?
A prokaryote.
Correct. So I see DNA, I see the replication bubble, I see a single strand coming off, so I know it's RNA rather than DNA, plus it's labeled mRNA, and then these ribosomes are attached. So this RNA is coming off of the DNA, so that's transcription. These ribosomes are attached to the RNA, making some chain of something, right? I know that would be amino acids, so that's translation. So now we're ready for our questions. So it was asking about which of the following best describes a characteristic of the process that is unique to prokaryotes. So it's only true of prokaryotes. So as you read these, if any of them are also true of eukaryotes, you have to rule them out. So which ones are we going to rule out because it's true of both?
B.
B is true of both. Do I build 5' to 3'? Yes, so that's true of both. So I'm down to C and D. Translation of the mRNA is occurring while the mRNA is being transcribed, or the enzyme that is transcribing the mRNA is RNA polymerase. Well, that's true in eukaryotes as well. So that means C has to be my answer. Now, yes, you might just know that, right? And you might have looked at that picture and right away been able to say C is your answer. But I'm trying to walk you through like the thought process. If you don't know, how can you make the best guess? Okay. I think that was the last one. Oh, I got one more slide. Did you know? I didn't. I just discovered this. So this is a student's test when I looked at the review. And the questions that you get wrong, did you guys already know this? They give you suggestions like this is topic 6.4. So I could look at the course at a glance and find out what topic that is, what skill is being assessed, 1A. I can look at that on my chart, but you guys have the, it's digital, the course description. But also, oh, you got that wrong. Maybe you want to review these videos. 6.4 video 1 and 6.4 video 2 will help you with this question.